Well, good morning and good afternoon and good evening to everyone. I am super excited today to moderate this great panel discussion for this very special webinar commemorating a very special time 25 years ago. My name is Pat Garcia-Gonzalez and I'm the head of the Max Foundation. I really want to welcome everyone and encourage you to write on the chat who you are, where you're coming from. It's very special for all of us to be united today to talk a little bit about an event that took place 25 years ago, and also, very importantly, to think about where we are with CML and the treatment of CML and where we're going for the next 25 years. And today I'm very honored to be in this panel with three very special people. I think Dr. Druker needs no introduction. And everybody knows his role, such a very important role in the lives of all people touched by CML. I'm also in the panel is Paula Boultbee, who maybe some of you don't know, but she played a very important role. She was the global brand director for Gleevec 25 years ago. And she was single-handedly responsible not only for the launch of the brand, but for the launch of this collaboration that the Max Foundation has had for 25 years to bring access to TKIs around the world. And then also many of you know Jan Geissler. I don't have to say much about him because he is somebody that has really changed the lives of people living with CML through his own experience, and he's one of the co-founders of the CML Advocates Network. So I look forward to this conversation. We will have time for questions, and please write any questions or comments that you want on the chat. So, I'd like to start by thinking back to 25 years ago. Where were we, what we were doing, each of us. And for us, we had started the Max Foundation. We had opened an email address, help@themaxfoundation.org. And one of the first messages that we received around this time was on behalf of a 22-year-old young man from Honduras named Raul, who was just diagnosed with CML and had heard about a new compound that was being developed, STI 571. As we know, it was the name of the compound. And he was asking us for help to access this treatment. And so we called Novartis, and the person that answered our call was Paula, in a way. And so that was the moment that sort of our lives interjected, and things changed, forever. We have been able to help more than 100,000 patients with CML after that, and provided more than 700,000 years of life. So Paula, I guess I'll start with you. What were you doing 25 years ago on this day?
On this day, my life was not, I was on a hotspot, and I would do one correction to Pat's statement is that there was a lot of people who were involved. So I would not single-handedly been able to launch a product like Gleevec out in the market. But I can say that I was one of the few believers in the Max Foundation from the beginning, because I was asked to do a patient assistance program. So I therefore lot of research found the Max Foundation would be a perfect partner. But what did I do in May, the 10th of May? I was actually on the launch meeting where we have all the countries collected in a hotel room, ballroom, where we were supposed to share information about Gleevec and the coming Gleevec. All of us were on our way to ASCO meeting, which was going to happen in San Francisco a few days later. And meanwhile, we are in this training meeting or sharing information meeting and making sure everybody understands what Gleevec is about. The approval came. So you need to remember that this is 25 years ago. The internet was not that you did go on a WebEx or you kind of got the video conference with somebody set up. So what we did something very unusual. We did a satellite feed of the approval from together with Tom Thompson, which was the health secretary at that time, and the CEO of Novartis, Vasella. They held a press conference, and that was transmitted to the room of all these people who are very eager to launch this product they have heard about from patients and from physicians, whatever. When is it coming? When is it coming? And rest of that is history. There's a lot of stories going around it, but it was really a remarkable day. Really remarkable day.
I can imagine. Dr. Druker, I cannot even imagine what your day was. What were you doing around 25 years ago?
Well, we obviously did a lot of interviews, but for me, it was kind of a bittersweet moment. And I've kind of compared it to sending a child off to college. And you have to realize I had been working on this project for well over 10, 12 years. I had been working in a lab for almost 20, and this was kind of my baby. And I had watched it grow from a very small, limited phase 1 clinical trial to massive worldwide phase 2 and phase 3 trials. And now it was going to get launched out into the world and available to everybody. And I was proud of that. I was pleased about it. But again, it did feel like I'm sending this out into the world to do some good work. And there was just also some private moments of reflection on what an amazing journey this had been, with so many people who participated from all the people at Novartis, all the clinical investigators, all the patient groups that came together. What an amazing, amazing accomplishment for patients with chronic myeloid leukemia.
Yes, and I was reflecting on the fact that technology also played a very important part. The fact that the development of this drug came about just about at the time of that the internet was playing such an important role. Mm-hmm. And Jan, maybe you can share your sort of journey and what were you doing 25 years ago?
Well, I mean, I got diagnosed 25 years ago, and it was actually before the approval of Gleevec in Europe. So it didn't look great. I was age 28 and wasn't sure whether I turned 29. And none of my first 4 opinions actually told me that there's something like STI-571 or Gleevec. So the 5th one actually told me. It was because I read about the data from the IRIS study from the US, and also read it on these early internet forums like Yahoo Asia and newcmldrug.com and CML Support UK. So there was a couple of grassroots initiatives. And then I wrote an email to some of the first authors, actually 2 of that paper of the studies. That was you, Brian Druker, and Andreas Hochhaus, and you were in a kind of competition answering super quickly, and gave me a bit of an insight what was going on in that space. And so I developed hope. There was no access in Europe at that time because expanded access was closed. So I joined a phase 1 study, phase 1/2 study with STI-571 and PEG interferon, which then worked very quickly. But there was no information in German language about this at all anywhere, and not even in the support group. So I was then starting to engage in translating ASH abstracts and your publications into German and putting that on blogs in the internet and Yahoo forums and whatever, to make sure that others can also understand. And I think it was not only, let's say, a revolution from a medicine's point of view, but it was because these grassroots movements from CML patients popped up in Israel, in the UK, in the US, in many other parts of the world, because everybody was desperate to understand, is this going to work? Is this a dream that we're seeing? Or is this just, after 6 months, we're going to see that it's been a bad dream? And so everybody was very excited. So it was an interesting time.
Yeah, very much so. Well, Dr. Radich, I'm so happy that you're joining us. We know that you're very busy at the Hutch, and appreciate you taking a minute to join us. What were you 25 years ago? I guess I've never asked you that question.
Yeah, I was likely in the lab, buried under all the BCR-ABL assays that we had to perform for these clinical trials. It's a strange story of coincidence. I mean, Brian and I actually went to the same undergraduate school at the same time, went off our different ways, and ended up back in the Northwest working on the same disease. And Brian probably doesn't remember this, but the whole reason I'm here today is because when the IRIS trial started, I was kind of buried in the transplant world doing BCR-ABL stuff because CML was a transplant disease then. And when the IRIS trial set up, Brian brought me into this to do the testing, and then from that, my life was changed.
Yeah.
Very, very much. I think the lives of so many people, were changed, and I can see the chat. People from all over the world are joining us from India and Rwanda and just Mozambique, everywhere, Nepal, everyone, because the story is also a story of access. A very unique story of access. And so it was really this amazing drug and this remarkable decision to ensure that people around the world would have access to it. And as we initiated the relationship with Paula and with Novartis to try to bring this drug to patients in 80 countries where otherwise, this drug was at the time not available. One of the biggest surprises that we had, something we really didn't plan on, was the reaction of patients. So we needed to make sure patients fulfill this criteria of this program, and then we would ask Novartis to send drug to the physician for that unique patient. And many patients started writing back to us and said, you just saved my life, and now I have to do something with the rest of my life, and you have to help me. And we did not expect that part. And so we started connecting patients with each other in countries and helping them establish patient organizations. And a lot of the work that Jan is doing these days is with so many people, so many patients that were touched by CML that were able to access this drug and decided to sort of change their life and help others. I think that's a very beautiful story that is sort of adjacent to the story of Gleevec. So I wonder, yes, Paula, yes.
I just like to add the complexity to the picture because I was in the middle of it, and Dr. Druker and Dr. Radich most probably are in the middle of it as well, in the sense that the day the drug was approved in the United States, it was not approved anywhere else in the world, right? So it was the first approval. So the clinical trial group has started a patient access program for the accelerated cases of CML. And patients around the world could be included in that one. And as the drug is approved in the United States, they said, okay, now patients have access to it. Here, Paula, here is this 4,000 patients. Convert them to something so they can continue the drug if they're doing well on the drug. And I was scratching my head, 4,000 patients, and they were everywhere. They were everywhere. Many of them were in the United States, which caused another problem, is that the drug is approved in the United States. They don't have the money to pay for the drug in the US out-of-pocket expenses. And their drug is not approved in their country. Many South American patients we had. So we needed to really do something. And that was even more urgent that we created this program with the Max Foundation, which those days were called the GPAP, to get all these patients converted to some free drug supply as long as they were doing well on the drug. But it was not only that. When they moved into their home country, whatever the country was, there was other patients who learned about it. And then they needed a diagnosis. And there was no diagnostic methods available. And they could do a bone marrow, but they didn't really have anybody who could sophisticatedly look at the bone marrow and say that, yeah, this is a clear CML case, so you can get the drug. Because that was one of the criteria to even get close to the drug in this GPAP program, that you need to have a very clear diagnosis, because everybody thought that it was a miracle drug, that it worked for every cancer, but it was not. So it was extremely complex, but we got the 4,000 patients off from the clinical trial or the patient access program to GPAP or something in between in those countries where we couldn't have GPAP.
Yeah.
And I'm saying GPAP because that's what hangs on to me.
And Paula, one of the things that Novartis did was, for the patients who are on the phase 1 and phase 2 trial, they actually were eligible to get continue to get free Gleevec. I bet you never thought that 25 years later, you'd still be sending
no,
patients.
Never.
Many of them are still on the program.
Yeah.
Many of them still on the program.
And it was very hard because you have these tragic newly diagnosed patient stories in the beginning, but we only had an approval for accelerated cases, and we didn't know where the drug was going. There were some adverse effects which came in very early days, and those scared everybody. Says, okay, no, stay with accelerated patients, don't get anybody in. But as the Irish studied that on the first line, patient come in, and you start to look better and better. Everybody's doing well on the drug. Now, what do? You can't close the door, but you can't really open it either, because you need to have the drug authorities or the FDAs, wherever they are in the world, to approve that indication for Novartis to stay out of trouble.
Yeah. And Dr. Druker, I guess did anything surprise you?
Well, there were so many surprises. And I think, as Jan and Paula had mentioned, and we had no idea how long this was going to last. And there was a part of me that thought, well, this is going to be like chemotherapy. We'll give it for 6 months. It'll work and cure the disease, or it will stop working. I never would have thought that we'd turn this into a disease where the majority of people are taking a pill a day for 25 years. That was not on my radar as something. And so that's been surprising to me. What I had predicted, even in 98, when I was writing about going to clinical trials, is I think this should work, but I have no idea how well tolerated it's going to be. And I realize that any number of people are struggling with some side effects, but compared to interferon, this was night and day in terms of its tolerability. But again, we had no idea. And that was something we couldn't predict. That's what clinical trials are all about.
Right.
But the other thing that was absolutely amazing, and both of you, you and Jan, have touched on this, was the rise of the internet. So in 1998, there were no chat rooms. Yeah. By 1999, I would go to clinic and I'd go back and I'd read about what I said in clinic, and we hadn't published anything. Nobody had heard about this drug, and people were in chat rooms. People were coming to the clinic. They had never met another CML patient before. And all of a sudden, people were banding together in these chat rooms, talking about what was happening, talking about their experiences. And this incredible community came together to make things, to give information about where can you get this drug, who can you talk to, what side effects are you experiencing, how are you managing that? This was completely new and unexpected.
Right. Do you want to comment on that, Jan? since you were a big
Yeah.
part of that.
No, absolutely. And I mean, everybody was nervous. And of course, we saw some people just took it lightly in the strictest meaning of the sense. And some people really had a hard time on TKIs. I mean, we can't deny that. Some people had resistance, some people had side effects. And I mean, the community was exchanging heavily on how to deal with that. And because this is a rare cancer, most hematologists also had no idea what this STI-571 or Gleevec was. They had no idea about management, monitoring, whatever. So it was often the patient community was pulling information from the publications, from the forums, and so on, and bringing that to the healthcare professionals and say, hey, I've read this, and this is how people dealt with rash, and that's how people dealt with whatever. So it was the patient community trying to get all the information, and that's probably what pulled the whole community also together, was the desperate need for information and for making sure that no patients left behind, not only with access to drug, but also access to proper management and best available knowledge. And I think that's what was an amazing interplay between the CML experts and the patient community and everybody involved, and of course the companies.
Yeah, I mean, another thing that was very surprising to me. I mean, I was coming, I'm not a physician. I was coming from our experience with Max years before, and the opportunity to bring this drug to patients around the world. Of course, we jumped on it and we started trying to figure out how to do this that we had never been done before. And it was 8 years into the GPAP program that I had a very important conversation with the hematologist from Ethiopia, who explained to me that yes, we were helping patients in the GPAP program, but that for the patients that were there, he had so many hundreds of others that didn't have money they didn't have a house to sell, to send their blood to Germany, to get themselves properly diagnosed so that they couldn't get in the program. And I thought this was a completely unacceptable, and that's something that we should change. And that's where I think my life and Dr. Radich's, because I started telling this story, and when I told Dr. Radich, he said, well, I think I've been working on something that might help you. And so I guess I'd love to hear from you, Jerry. What surprised you? What has been the story of the development of diagnostics and how the drug changed how we look at the monitoring, the diagnostics of CML?
Sure thing. So, I mean, my first surprise was along the lines of Brian's, because I had been studying CML progression, and I thought, well, in the end, Darwin always wins, right? So there's going to be some way that this disease is going to get around this, right? So when it didn't, that really had a big implication about monitoring, because it meant we had to kind of do long-term monitoring on patients. And I think one of the surprises was how well it worked, the monitoring. B, how even regulatory people accepted this. Right? So we were able to turn overall survival 10-year studies into very brief studies based on the molecular markers, and they got FDA approval. And that was fantastic. And then to your point, we had a couple of lucky circumstances that got us to be able to really kind of refocus our lab on ways to get diagnostics into these patients who needed it and around the world. I can still remember the first time that Pat came to the lab and looking for these Cepheid cartridges that we had kind of invented, worked with Cepheid in the lab to develop. We had people from Cepheid embedded in our lab. And Pat looking at the box of cartridges and having sort of a tug of war with my main tech about who was going to get the cartridges, because we were going to give her 10 and Pat wanted 200. So it was just a tug of war, which says a lot about Pat. Persistence.
Yes.
That literally that moment kind of changed the whole part of our mission in the lab, and it was really kind of life changing for a lot of us.
Yes, I mean, I think that that story of bringing diagnostics, I really did not realize what an important piece that was. And also, over the 25 years, what I find that sometimes is so remarkable on the arc of the message is that I think for the first 10 years or so, we were told, and we needed to tell patients, you never can skip this drug, and you will take this drug for the rest of your life. Okay. And we pounded that was pounded on us. We told patients, you can never, ever, ever, ever stop this drug. And then one day, somebody said, well, turns out maybe you can stop this drug. And it has been difficult to shift that messaging
Yeah, sure.
patients. And
Mhm.
I imagine that seeing a patient that is newly diagnosed today and what you're telling them is very different from what you were telling patients, 15 or 20 years ago. And for a lot of patients, and I can see the chat, many are here with us. How do we explain the change between you will never be able to stop this drug to, well, some people perhaps can stop, and how do you make that in a safe way, Dr. Druker?
Well, it is so interesting, and I'm so grateful to the French group that pioneered treatment-free remission. I had actually proposed this to some of my early patients whose PCRs were undetectable for several years, and they were like, "No way am I going to ever stop this drug. It saved my life. I was dead without this." And I have some patients who've been undetectable from the phase 1 trial 20 years or more, and they still won't stop. And some of the patients that get diagnosed today, I know I can start the conversation with, I'm confident you're going to live a normal lifespan from your leukemia. You'll probably die of something else. But then the conversation is, how long do I have to take this medication? And then we begin to talk about, if the goal is to get you to undetectable, then we might be able to stop therapy. For other people, the conversation is more around quality of life and trying to lower their dose to the lowest possible dose that maintains their leukemia in an optimal response. But that's where we have to engage our patients and set our goals and try to figure out what's going to be best for each individual. But I'm confident that I know I'm going to be able to keep my patients safe. And I know I'm going to be able to keep them alive, because the numbers of people that will progress to an accelerated or blast crisis are minimal. I send one patient out of my 500 or so to transplant a year these days. And, you guys thought we were going to put you out of business, but you've done pretty well in the transplant world. But it is a very different conversation around quality of life, around goals, and engaging patients in those conversations.
Yeah, exactly. And I mean, let's just shift a little bit to everything is not solved, right? A lot is great. I don't think there's been access to any other innovative treatment in the past 25 years, like there has been access to imatinib. There's also some access to second and third generation. We still continue to increase access to monitoring, but there are still entire countries that are not able to do PCR, and still we need to send the blood. We do this with Dr. Radich's lab. So important work, but there's so much to be done. So perhaps we can shift a little bit to see what are the barriers. And Paula, you have been now, after leaving Novartis, in the board of the Max Foundation, that I have feel very honored to be with you, because you're such an important part of the story. So through our work what the barriers are to access. So maybe you can speak from your perspective a little bit, what most concerns you at this point.
I mean, my biggest concern is not that we can ship drugs safely today. We can organize with the vendors to get the drug from the warehouse to a physician who can give it. But what bugs me a lot is the diagnostics. It's even in the United States that the standards are missing, the monitoring of these patients are missing, that nobody seems to be able to get together. What is the PCR value I'm looking for? What is the no detectable? Because it's depending on the machine, which machine you use and which assays you use and all that. And as soon as you leave the United States and then mainland Europe and maybe Japan and some other countries, it's almost non-existent. So the biggest hurdle, I think, for good access for patients, even those patients in low middle income countries who can pay for the drug themselves, is to get the proper diagnosis. And it's not only CML, it's many, many other cancer diseases, but particularly in CML, where you do have a very excellent drug, you could help these patients for lifetime or help them to get off the drug. But and I really struggle with it. What could you do? Do you need to start another foundation to try to get diagnostics, or do you need to start an education group? What do you need? We did a lot during Gleevec. We developed assays for GIST, and we did introduce PCR working groups and try to get physicians together. And it worked in some countries. Meanwhile, it didn't work that well. But we did not get the big diagnostic companies even 25 years ago to develop assays for CML. They have been a little bit better, but I still that the diagnostic industry could potentially be more forward and simplify them. And I don't know, as a commercial person, I would not say this, but be less competitive on the assays and the machines, because that's what limits people's access in many countries, that one hospital buys this machine, therefore they can't use this one, or vice versa.
Right.
So that would be wonderful news if we could maybe isolate even countries to get better diagnostics to them.
Jerry, I guess, are you going to save us and develop?
Jerry, please save us. You have saved us many times. I think you can save us this time.
I mean, there are things that we can do. We're working on some low cost point of contact assays. I mean, we've developed a lateral flow device that's like a pregnancy test for the 359 mutation. We're developing those for CML and for PML-RR, which would be a rapid test that you can do just like at your home or whatever. We've got ways to do, blood draws out of your arm that we can use for spot blood spot diagnostics and stuff, so that places that don't have syringes and the like can get access. And you can imagine in a world where there are drones and cell phones, there's no reason that people can't summon a drone with a blood test that you can do and then send the thing back to a central lab. So I think there are solutions. I think we have to kind of we have to use the work in low and middle income companies as a way to leapfrog the current technologies that are embedded here, right? I mean, in the end, we can't develop for other diseases. We can't develop cytogenetic labs and the like. We've got to have niduses of sequencing, right? And just leapfrog, take this as an opportunity to leapfrog over the kind of prevailing technologies in the weird countries and use it as kind of an opportunity.
Right.
But Jerry, there's two problems you've got to fix. One is access around the entire world, as I think you've outlined some there. But even in the U.S.,
oh yeah, absolutely,
the PCR testing is not very well standardized. And if our patients go to different laboratories, they'll get different results, and then I've got to figure out, okay, if they lost their response, where do we reach? Yep. How do we make sure they get them checked at the same lab every time, so we can actually monitor trends over time? And I've just been unimpressed by how the U.S. has been unable to standardize this type of testing.
Yeah, no, it's it's I think a number of factors. One is that the Western medicine system is always sort of cowboy medicine, kind of everyone's on their own. it's a real problem with groups and clinics renegotiating contracts with different companies to get a better deal. And so I think a lot of the community docs don't even know where to send it from year to year, right? It gets changed all the time. And like you said, even though we've tried to do the international scale, the international scale does not save a lousy test. So there's that as well. But yeah, but you're right. But I've got people in Montana that have to drive 6 hours for a PCR test. I mean, that's kind of ridiculous, right?
No sense. Yeah.
Yeah.
Yeah, and from the advocacy perspective, it's such a different time now, 25 years later. I feel like the urgency and the drive that drove patients to pursue, access to TKIs 25 years ago and to start patient organizations, I fear that's kind of not there. So, what are you doing in the advocacy community to make sure that there's a new crop of advocates that continues to advocate for people living with CML?
Well, I mean, of course, we're at the maturity in terms of therapies that the urgency, if you have access to therapy and diagnostics, let's just assume that, which we know is still the biggest bear and biggest problem worldwide. But if we just assume that patients have access to all current TKIs and inhibitors and so on, we just need to acknowledge it's not a cure. I mean, we've invented terms like functional cures and stuff like that. I've always opposed that a bit because I feel just the absence of rising counts doesn't call it a cure. There needs to be some biology with it. And we need to face the fact that 1/3 of patients, even in the best access scenarios, can actually stop treatment, which means 2/3 can't. And that actually means we need to deal with a chronic cancer, needs to be treated lifelong. You need to do PCR every couple of weeks. And you need to deal with probably low-grade side effects, which can be really annoying. And I think that drives people, quality of life, fertility, trying to do family planning, trying to, let's say, have a social life and so on. Some people suffering heavily still, even though it's a small proportion. But I feel like the topic of cure is still something that really drives the community. So in CML Advocates Network, we've held 2 academic community advisory boards where we didn't invite the key clinicians that drove all the TKI studies, but more the people that do modeling, look at academic approaches to cure, new scientific methods, is CAR-T an option, cell therapies, whatever, gene therapies. And of course, it's super difficult to develop a therapy in the setting where the comfort level is so high that you would probably not take a very toxic treatment. But I feel still those that say that's not the end of the line, it's not the end of the story. We need a cure and we need to get rid of CML finally. That story is not over. So I think still science, let's say, activates advocates, even though it's different than the motivations we had years ago. That's true.
Yeah.
Yeah, exactly. I mean, there's so much. When I hear about CAR-T, I think we're crazy. I think we're going back to transplant. Like that cannot
Yeah.
be, that cannot be the solution. But it's true that, one of the other surprises was now that we have reached more than 100,000 patients in low and middle income countries, is the median age of diagnosis, which is 36 years old. In the world, this is a disease of young, productive people and many, many children. Last year I was able to go to Guatemala and to El Salvador, and in both places I met a little girl. The one was 5 and the other one was 7. Both of them diagnosed at age 2.
Wow.
And I think about them when I think about their life and the parents. Like, what are we doing for these children? Because now they have their whole life. Are they going to have to take this medicine? What's in the horizon for them? So it does, personally, that does keep me up at night sometimes to to think about patients that need long-term treatment. So I agree with Jan, and I'm very glad both Jerry and Brian, you're both involved with the International CML Foundation and the work towards a cure, because we do need to make sure all patients can safely stop treatment and cure their CML. So let's just take just 1 minute to talk about what's exciting, maybe in that area, and then we'll open up for questions. So do you want to share, Dr. Druker? Are you excited?
Yes, so thank you, Jan and Pat, for mentioning the International CML Foundation. And Jan, yes, we've heard you, and we agree that curing this leukemia is something we absolutely want to do. And it's our view that we have to take the same approach. With imatinib for CML, we understood what was driving the growth of leukemia, and we targeted it. Now we have patients who seem to have a pool of persistent cells, and if we could figure out what allows those cells to persist, is there a way that we could use that as a target to eradicate those residual pool of cells? And to make that work, it has to be effective, but it also has to be safe. I mean, I'm not going to put somebody through a highly toxic therapy for a cure that's going to leave them with a different long-term chronic illness. So it has to reach a pretty high bar in terms of something we know that mostly will be effective, but also has to be really safe. But it has to be based on knowledge. We have to understand, what is it about those cells that allow them to persist? And if we understood that, that gives us a target to eradicate it. And that's what the International CML Foundation Consortium is going to work on.
Great.
Yeah. And Jerry, what excites you about the future?
Well, I mean, I'm with Brian on this. I think that there's a ways to go to kind of understand the difference between great responders and lousy responders. And, that looks like stuff we've done looks like it's mostly inflammatory and immune situation, right? So we did some stuff, and 20 out of 20 top pathways are all immune regulated. So there's something there to go for. And I think that the idea also of, looking at areas in low and middle countries where we can go in and think about discontinuation for these people who have been on drugs for years and years and years, because there's only so much drug to go around, right? So if you could take it from patients who actually don't need it and then find new patients, that's going to be a huge impact. So I think the exciting part now would be sort of to map the strategy of how you kind of come into a community that hasn't had much testing, get an idea from a couple points of testing, which are going to be the people to safely do that, and use that kind of algorithm to drive more sustained discontinuation efforts. I think that would be really fun to do.
Yeah, definitely. I do think a lot about that, because we have about 30,000 patients on drug at the Max Foundation, and I think it's about 20,000 of them that have been on treatment for more than 8 years. And I'm thinking, I know there are people that can't stop treatment, we just don't know who they are.
Yeah.
And so, on the one hand, we want to make sure if somebody doesn't need the treatment, that they don't take a treatment that they don't need, at the same time that we are trying to make sure everybody can have access to the treatments we need. So there's a lot to do. But I just want to, before we go to questions, I just want to acknowledge also the dedication of all the hematologists around the world that are treating CML patients. And some of them who have 1,000 patients under their care. And I've been on the CML Day clinic day, where there's 150 people waiting to be seen in the hospital lobby. So The dedication, I know I just saw Dr. Malhotra from India is on the call, Dr. Silva from Mozambique, who has fought for CML patients, for 20 years. I just want to acknowledge all the hematologists and the work that they do every day for patients. I think that their dedication inspires me to sort of keep going and trying to make sure that no patient goes without treatment. So Jeff, what's next? Do you want us to open to questions? Are you going to?
That would be great. Yeah, we have a few in the chat. I'm going to jump to one now. Pedro's asking about, is there any work going on to improve the existing drugs, like imatinib, and make them better? I don't know who wants to take that. Dr. Druker?
Well, I'll talk about some work actually to improve dasatinib. And there's a group in Sweden, for patients who are taking dasatinib or Sprycel, there's some interactions with some H2 block, the anti-heartburn medications. Their effectiveness isn't quite as good if you take them with those medications. This group in Sweden has developed a formulation that allows you to take them with the medicines that would help your heartburn. So those are some of the things that are coming along. And Novartis has had 25 years going from imatinib to nilotinib and now to asciminib. It has opened up a whole new way of targeting BCR-ABL. There's at least one other kind of a knockoff of that drug in clinical trials. There's a drug from a company called Enliven, which is a kind of a different drug than ponatinib that's dialed out some of the serious side effects from ponatinib that's making its way through clinical trials. So the reality is, 5 or 6 different drugs approved, 3 or 4 more in clinical trials. That's pretty remarkable for what was once considered a rare disease where you might not make a profit on drug development in this disease.
Next, I have a question from Kamal about, after curing CML, can we continue imatinib? I'm thinking it's probably more around TFR. Jerry, you want to jump on that one? I'm trying to formulate what actually the question is actually asking. Why don't you try that again? Let me try that again. It's asking basically, after curing CML, can you go off imatinib? But not much beyond that. I don't know if the person can add some more to the question in the chat. But I was thinking they were thinking about TFR and going off therapy and what that requires. Well, we know that in tons of studies. So again, led by the French group, that if you've been in a deep molecular response for a number of years, that you've got roughly a 50-50 shot of getting off drug without molecular reoccurrence. And if you stay on drug longer, that odds of being able to stay off drug improves, which makes some sense.
Right.
And so, I think as Brian was saying, to optimizing your care for every patient. One of the things that's remarkable in this drug, we have a really good marker of disease burden, right? So we should never be in a situation, if we have access to diagnostics, of being really too concerned, because if you monitor people and their disease is coming back, you'll know it. It's not like you don't have to do bone marrow. It's not like an acute leukemia. We have to wait and do another bone marrow. You can easily tell, if people are going in the wrong direction or not. So I think that we have all the tools to be able to really optimize therapy and response on a person-to-person basis.
Yes.
Jared, out of curiosity, Jared, sorry, are you impressed, or are you using any of the tools to try to figure out who could predict more than a 50-50 chance? And are you using digital PCR? What's your thoughts on, can we do better than 50-50?
Yeah. The better tool, the digital PCR, is a little bit about a half a log more sensitive. I think that helps a little bit more on the margins. So far, the biomarkers have not been very good. We with Novartis spent an enormous amount of taxpayers' money definitively proving that gene expression at the time of discontinuation and beyond that is not a good biomarker, right? So we missed out on that one. So I think a lot of people are in the hunt, but so far we haven't found the real secret. Again, a little bit more sensitive assay. We're developing assays that get down to 1 in 10 to the 7th, so that might be the trick. But we're not quite there yet.
Thanks. Just want to make sure I wasn't missing something.
You're never missing anything.
I just want us to give a reality check to patients because that person in Nepal is asking if he can stop treatment. That's what he was trying to say. And I just want us to say very clear to patients, there's two very different messages. Very exciting, everything that's happening. For each patient, first of all, make sure that you're taking your drug every day. Make sure you have access to monitoring and that you have good numbers in monitoring and that you will be able to monitor your disease if you and your physician decide to attempt to stop treatment. And that's a part that sometimes is not quite heard by patients. And I know so many people then go stop their treatment and don't tell their physician. So let's not do that.
Yes, but there's a couple of really interesting things here, Pat, that also surprised me. And so the first surprise was I thought that if somebody stopped there, had been undetectable for 5 years, stopped therapy, and they recurred, that I need to get now 10 years of undetectable before I could try stopping therapy again. In fact, they get back to undetectable, you wait 2 years, undetectable. They have a reasonably good chance of being able to stop therapy on the second chance.
Yeah,
and
that shocked me. That shocks me completely. Yeah.
The other was that I have a number of patients who've stopped therapy and I have this one patient I showed his PCR. He's been off therapy for 15 years now. And in 15 years, he's had 3 or 4 undetectables, and everything else has been between 0.003 and 0.01, but never above 0.1 on the international scale. And clearly there's something about, I don't know, it's the immune system, what it is that's allowing him to control his leukemia. But it's also led to studies where people with very, very low level PCRs, like 0.005, are able to stop therapy. So it doesn't have to be perfectly undetectable. And again, that was totally surprising to me.
No, if I was a betting person and bet on some of my predictions about this whole process, I would have been broke a long time ago. And I would have just completely lost the farm on many occasions.
And the points you raise are really important in the sense that one of the areas that we're moving forward and that Pat and our whole team are talking about a lot is how to optimize care for these patients, which starts with, at minimum, at least an annual PCR. And it's something that we're working hard to, you'll see later in the year around World CML Day, we're going to start to push this message, which is really not a low income message. It's a global message for CML care, regardless of what country you're in. And it's just something that has to happen, whether you're in Des Moines or in Nairobi. You need to get regular testing to make sure that you're having adequate response. And only with that can you figure out if you have the opportunity to stop treatment, or maybe you need another med because the one you're on isn't doing what it needs to do. In the chat, actually, we had Dr. Arena, Dr. Patel, and Essa all ask about, Pat, other drugs and what we're doing to try to get access for CLL patients in Georgia, for patients in Gambia, the continued need to move beyond imatinib.
Yeah, I mean, I'll say, the time that we are 25 years later, many of these treatments are now generic. There are generic versions of these of TKIs. And so it's a whole different thing for us to try to figure out at what point can this treatment be adopted by the local governments and ministries of health and the health systems of these countries, so that we can continue to provide the drugs that are on patent but the transition to a time when each country has access to these drugs is now. And I think that for the advocacy community, Jan, when we're going to ministries of health to talk about CML, many times we're told CML is not a priority. And I think that we need to think together with the CML Advocates Network, how can we make sure that CML is a priority in every country in the world, and that we start taking steps towards a sort of long-term availability of these drugs in every country in the world. So, as the Max Foundation, we're committed to continuing to provide new drugs as long as they're not available. But we are at that time when many of the TKIs are now past the loss of the exclusivity of the company. And now we are, as you know so well, I don't have to tell you, we're in the era of like, how do we figure out the quality and affordability of these drugs? These are very important points. You want to comment, Jan?
Yeah, probably just one thing. I mean, CML has always been a pioneer disease. We've been pioneers in the beginning in the development of the pathways. We've been pioneers in bringing access to low and middle income countries. We as a collective, and everyone in their own role. We've been pioneers in dealing with chronifying a cancer and so on. And I feel policy change can often not be done just on the disease level, because there's usually no CML law in any parliament and no exception just because of a specific disease. It needs to be policy change. And that's why I've always followed with my advocacy work, trying to get different rare cancer organizations together. You've done that, Pat, in the Max Foundation with all your team as well, looking at different diseases using the same mechanisms and showing this is systemic. It's not just CML or breast cancer or PNH or whatever. And I think that's what we need to strive for, because we have so many challenges in access, in regulatory pathways to even approve new medicines that might be a cure, and to also get through cost effectiveness discussions. It's all very technical, but it only works if we speak the language of policy and regulators, and trying to convince them that trying this out with CML will bring about systematic change, that other patients benefit also in other rare cancers or beyond. And I think that's probably something for us as a CML community to think of. How do we do alliances? How do we collaborate on these things?
Great, thanks, Sean. We're coming close to the close. We have multiple questions from Bahija and a few others around trying to understand how monitoring can lead to understanding who's going to have resistance, whose disease is going to be resistant to treatment, and whose is not. Which Jerry talked about the biomarkers. I don't know if Dr. Druker, Dr. Radich, you want to talk about the challenges there.
If we could understand that, I know there are a number of comments in the chat about how can we figure out who's going to successfully be able to stop therapy versus who hasn't. We've been working on that in our lab and Jerry's and others for quite some time. And if we could figure that out, we certainly could get more people to a successful TFR, and we absolutely are committed to making that happen. I also just wanted to comment, I saw a number of comments in the chat about gastrointestinal stromal tumor, and huge shout out to the groups that have benefited from imatinib when it got extended to that disease. Work that actually started in my lab and then was extended to groups in Boston and then to the clinical trials, and just another fabulous success story of remarkable turnarounds and people now here alive and thriving. But a huge shout out to everybody at Novartis that was on board for this program, and especially the Max Foundation. What an incredible job you're doing. As Jan said, truly bringing global access to a life-saving medication, and what amazing work, and what a great example of what can be done when people come together to collaborate for the benefit of people around the world.
Yeah, I would just like to double down on that, Brian. I mean, this is a collaboration that's truly unique between, a nonprofit, biotech, pharma, and academia. I mean, I don't know of any collaboration out there that has brought those together in really a purposeful way. And I think it's a fantastic, good news story in times that need good news.
Yeah.
Mhm.
Yeah, and the remarkable thing when I had the privilege to visit some of the hospitals with some of the physicians is there's hundreds, if not thousands, of people that we don't know who they are, that somehow step up to make this happen. This doesn't just happen. There's so many layers to logistics of keeping 40,000 patients on treatment all the time in 75 countries around the world. But it's done by pharmacists and nurses and custom clearance people. And there's also the patients that there's a patient in Chiapas, I think, in Mexico, Jerry, that he goes and he brings the other patients together so they can get the blood to put on the cards to send to you. But it's like so many people that step up and do the right thing and make this happen. So, I mean, I think it is a remarkable thing. I feel very privileged and lucky. And we need to keep going. So I'm here for the next 25 years. I think we need the young people to join us next time, because we need to keep.
Yeah.
There's so many thank yous in the chat. It's remarkable. We have patients talking and expressing their gratitude and thanks. I'm going to say to Sarah, I think we need to look up the date of approval for imatinib in GIST and have a similar teleconference with a slightly different cast to talk about that moment, because it's a huge moment. So we're at time. So Pat, I didn't know if you wanted to close and give each for our panelists a final word.
I'll just let Paula, we haven't heard from her in a minute, so you can close it out.
So you make me to close the meeting. This is fantastic. I think it's wonderful to see that it continues, that nobody is stopping the development of CML drugs and understanding the disease. Because when you started 25 years ago and it was such as meniscus-related disease, nobody really cared about it. But of course, I think we had a CEO at Novartis at that point in time who really put his head to it and said, this needs to work and we're going to move forward. And most probably he had a lot of insight from Dr. Druker and others around in the world. So he was a bigger believer than anybody else. But from the commercial point of view and from all the benefit to the patient, this drug has been given for 25 years. And here that we are continuing the mission and continuing to find better diagnostics and better drugs and understanding the disease, it's wonderful to hear. Absolutely wonderful. And I don't know if I promise the same thing as Pat to be here for the next 25 years, but if I will celebrate my 100th birthday, maybe we'll be in the podcast with the Max Foundation. So thank you, everybody, for being here, all the physicians, all the patients, and all the doctors around the mission.
Thank you so much.
Thanks, everyone.
You're welcome. Good day, everyone.
Thank
Thank
you.
you.
Thank you.